So first, we will discuss the definition of enzyme action. What is enzyme action? An enzyme attracts substrate to its active site, catalyzes the chemical reaction by which products are formed, and then allows the product to dissociate, separate from the enzyme surface. The combination formed by the enzyme and its substrates is called the enzyme-substrate complex. So next, we will discuss the structure and function of enzyme. Enzymes are catalyzed. They are usually protein, though some RNA molecules act as enzymes too. Enzymes lower the activation energy of a reaction, that is, the required amount of energy needed for a reaction to occur. They do this by binding to a substrate and holding it in a way that allows the reaction to happen more efficiently. The part of the enzyme where the substrate binds is called the active site. Here, the enzyme changes shape slightly, fitting tightly with the substrate and forming the enzyme substrate complex. Next is the factors affecting enzyme activities. Enzyme activity can be affected by a variety of factors such as temperature, pH, and concentration. Enzyme works best within specific temperature and pH ranges, and suboptimal condition can cause an enzyme to lose its ability to bind to a substrate. The first factor affecting enzyme activity is temperature. Raising temperature generally speeds up a reaction, and lowering temperature slows down a reaction. However, extreme high temperature can cause an enzyme to lose its shape and stop working. The second factor that affecting enzymes activity is pH. Each enzyme has an optimum pH range. Changing the pH outside of this range will slow enzymes activity. Extreme pH values can cause enzymes to denature or stop working. The third factor is enzyme concentration. Increasing enzyme concentration will speed up the reaction, as long as there is substrate available to bind to. Once all of the substrate is bound, the reaction will no longer speed up, since there will be nothing for additional enzymes to bind to. The fourth and the last factor that affects enzymes activity is substrate concentration. Increasing substrate concentration also increasing the rate of reaction to a certain point. Once all of the enzymes have bound, any substrate increases will have no effect on the rate of reaction, as the available enzymes will be saturated and working at their maximum rate. And now, we will discuss the common mistake and misconception of enzymes action. First, enzymes are specific. Each type of enzyme typically only reacts with one or a couple of substrates. Some enzymes are more specific than others and will only accept one particular substrate. Other enzymes can act on a range of molecules as long as they contain the types of bond or chemical group that the enzymes target. Lastly, Enzymes are reusable. Enzymes are not reactant and are not used up during the reaction. Once an enzyme binds to a substrate and catalyzes the reaction, the enzyme is released unchanged and can be used for another reaction. This means that for each reaction, there does not need to be a 1:1